Welcome back to part 2 of chapter 2. So we're going to talk about darkfield microscopy for just a moment. The darkfield microscope is a bright field microscope that has a modification known as an opaque light stop. You can see it right here. What this does is the light comes up, that light stop causes a refraction of the light. Basically, light is blocked in the very center, and it only comes around the edges. Once it hits the condenser, then it's going to pass through the image, but the only light that actually hits the objective lens is the light that reflects off the images or off the specimens that you have. And so they end up looking very light on a dark background, and that's dark field microscopy. So what this allows is for us to identify the structure of microbes without actually staining them or killing them. In phase contrast microscopy, we use an annular ring and a phase plate to produce a high contrast image without staining. And so the annular stop, which contains the annular ring, produces a cone of light that travels directly from the illuminator source to the condenser, but that cone of light is reflected off of that annular ring. The objective then contains that phase plate, and the phase plate contains that phase ring. So light traveling directly from the illuminator passes through the phase ring, while light refracted by the specimen passes through the phase plate. And so you can see light that has been diffracted and light that was not diffracted, depending on where it came through. And this produces that contrast that you see. So here you don't see as much. This is what bright field microscopy will look like if you're looking at a simple squamous epithelial cell, like a cheek scraping. And here you can see that same image under a phase contrast microscope. You can see the nucleus much more effectively. You can even see structures that are likely bacteria that are found within the epithelium. There we go. In fluorescence microscopy, we're again using stains or fluorochromes to allow us to see the microbes. And so this is really important in clinical microbiology when you're trying to identify certain pathogens that are causing diseases. So there's a very important test known as immunofluorescence, which allows you to identify certain disease-causing microbes. And it does so by taking antibodies that contain a fluorochrome that will fluoresce, and then having those antibodies, allowing those antibodies to adhere to the pathogen. And so in direct immunofluorescence, the fluorochrome actually attaches directly to a primary antibody. That primary antibody attaches to the antigen. In the indirect immunofluorescence, the fluorochrome attaches to a secondary antibody. That secondary antibody is going to attach to the primary antibody, which will then still attach to the pathogen. And so this is one mechanism we use to identify rabies virus, the rabies virus in the system. If you're going to be moving up in biology, in microbiology, then you may get the chance to use an electron microscope. Electron microscopes are much more powerful than our bright field microscopes that you're going to be using. So with bright field microscopes, we have a maximum magnification of 1,000 times the image size. So if the image is, 1 micrometer, then we're going to see it as 1,000 micrometers. Make sense? Some structures are too small. They're smaller than that, and they are so small that you can't see them. And so that's where electron microscopes become really important. So what electron microscopes do is they can magnify and make a very sharp image of up to 100,000 times the size. So not only can we see the cell, but we might be able to see the DNA within the cell or the organelles perfectly. Or in bacteria, we might be able to see their inclusion bodies and their ribosomes really effectively. There are two basic types of transmission or of electron microscopes, the transmission electron microscope and then the scanning electron microscope. Transmission electron microscopes produce an image that is far more powerful than the bright field microscopes, but the concept behind how the image is produced is similar, except for they're using electrons. Bright field microscopy is slightly different. Scanning electron microscopy forms a three-dimensional image of the structure by beaming electrons to the cell surface or to the structure, I should say, and knocking those electrons into that surface. That actually produces a 3D image, so you see more of a structure versus that flat 2D that we normally see when we're looking in our bright field microscopes. So on page 69 of the textbook, you have a table. It's called a figure, but it's really a table of all the different types of microscopes and their typical uses, and then it even shows sample images for each of the types of microscopes. We didn't talk about every single type of microscope. We talked more about the most commonly used microscopes, okay? And now we're going to get into section 2.4, which is staining. This is the fun stuff. So once we get into staining in the lab, you're going to start having a lot more fun in lab. That's usually the part where everybody just falls in love with microbiology. I'll never say that the theory is the most exciting portion of this class. It really is the lab that makes everybody love it. And so we use light microscopes, and when we use light microscopes, we have to stain the pathogen, or I shouldn't say pathogen, the microbe, because not all microbes, like I've told you in the past, not all microorganisms or not all bacteria are pathogenic. But we have to stain them so we can see them. They are invisible to the naked eye. So you can make a wet mount, or you can do a dry mount. I'm going to focus on dry mounts because that's what we do in our lab. But a wet mount is something that you might do if you want to see living organisms in, say, pond water, so you can see how they move and what their structure is. What we're going to look at is fixing a specimen and then staining that specimen. So with wet mounts, we don't tend to stain as often because that's not the purpose. We just want to see what they look like under the microscope. With bacteria, we're not going to be able to use a wet mount. So we fix the specimen. And so you can fix bacteria in different ways. Either the specimen is placed on a slide, which I'll talk about, and then is heat fixed, which is what we're going to show you, or what I will show you in lab, and what I'll talk about here as well. Or we can use chemicals on the specimen that fixes them and kills the specimen. So when you fix the specimen, either with the use of chemicals or heat, you're killing that specimen. So the bacteria are dead when we actually look at them under the microscope. And so we use heat fixing to fix the slides. How we do this is we have a slide, you can see it right here, and we're going to run the slide. We're going to aseptically transfer bacteria to the surface of the slide, and then we run the slide through the fire of a Bunsen burner. We do this to dehydrate the slide as well as fix the pathogen, or I keep saying pathogen, I'm not meaning pathogen, fix the specimen to the slide. So then it's basically stuck on the slide, it's not going to come off the slide. If you forget to heat fix the specimen, then when you're rinsing your stain off of the slide, the bacteria will also be rinsed off and you'll have nothing left. So you always have to remember to fix the specimen. Once you fix the specimen to the slide, then you're going to be able to look at them under a microscope. But the only way we can do this, bacteria are invisible, so you wouldn't see anything. You might see a cloudy image on the slide because there's something there. It's, you know, dirty now. But you can't actually see the cells. So what we have to do is we have to stain the cells. So there are two different techniques or two different types of stains that we tend to use. Bacteria themselves are negatively charged, so we can use a basic dye, which is positively charged, and it will stick to the bacterial cells. Positives and negatives are attracted to each other. This is true in biology and in chemistry. Opposites attract. We can also use acidic dyes. Acidic dyes are negatively charged, and they will be repelled from the bacteria. And so they don't stick to the bacteria, they'll stick to everything but the bacteria. And in this case, if it's a negative charge, then the bacteria sit clear on a dark background. It kind of looks like a darkfield microscopy, but it's not. It's called a negative stain. Then we have a gram stain, which uses more than one dye, an acid-fast stain that uses more than one dye and a major different technique, and then endospore stain. So we're going to talk about each of these. So for the first one, in a simple stain, like I said, bacteria are negatively charged. And so in a simple stain, we use that basic dye, which is positively charged. Here we have the culture is spread over a slide. And this is going to be either from a broth or from an agar plate. But you put the bacteria on the slide. You can let it air dry. I typically don't have patience for that, so I take that slide and then I use a clothespin because I don't want to put my fingers too close to the fire, and then you run it through the flame. And you do this slowly, and you make sure that you have enough time, because if you have glass over fire for too long, do you know what happens? Giving you a chance to think about it, and I'm going to answer it now. The glass will pop, it will break, and then you've just lost your slide. So you always have to be patient. Run it through the flame, let it sit out of the flame, run it through the flame, let it sit out until it is completely dehydrated and dry, and that will not only set the bacteria in place, it fixes it, but it also kills the bacteria. We then flood the slide with that basic dye. So this basic dye could be something like safaranin, it could be methylene blue, it could be crystal violet. We let it sit for about a minute to 2 minutes. We then rinse with distilled water and then blot it dry using bibulous paper. Once we're done, we look at it under that oil immersion lens. We always, when you use a microscope, you always start on the lowest power scanning, get it focused, move to the low power, get it focused, move to the high power, get it focused, add a drop of oil and move to the oil lens, and then focus it one last time, and then you'll see your specimen. And again, I will show you how to use the microscope before you're actually using your microscope, okay? In a negative stain, so this is also a simple technique, we use an acidic dye. The one that we typically use is called nigrosin. Acidic dyes are negatively charged, so they're going to be repelled because opposites are attracted to each other. The same is repelled. Think about magnets, pushing them together. If they're the same side, they're not going to want to go together. And so what happens is we have a dark background with clear cells. So in this mechanism, we take a simple dye, or not, we take our bacteria, we put it on a slide, but we put it on one side of the slide. We add nigrosin to the bacteria, we mix it up, so nigrosin is the actual stain, and then if you've ever done a smear, a blood smear or a bacterial smear, that's what you're doing. You're going to take a clean slide, you're going to move the slide at about a 45-degree angle into the specimen where the bacteria and the dye are mixed together. And then you just spread it all the way across, so you're making a smear of it. This slide here is now contaminated, and so we throw it into the biohazard container. This slide here, we're going to let sit, and you have to let it sit until it's dry. And so that can take anywhere from 5 to 20 minutes, depending on how much nigrosin and fluid was on the slide. Once it's dry, then again, you're going to focus the microscope, get it to the oil immersion lens, and look at your specimen. There's only like 3 slides. I know this is already 17 minutes. I'm just going to finish up relatively quickly. So this is just showing, again, a simple stain and a negative stain, which we already talked about. So this is just your figure showing those structures. This is what you can tell on a simple stain is the structure and the size, but that's all you can tell on a simple stain. On a negative stain, you cannot tell anything else about the bacteria. You can tell the structure, so rod-shaped. You can tell the size, so streptobacilli, relatively large rods. That's about all you could say here. A gram stain is a differential stain technique. And so in a gram stain, we're actually going to be able to see the difference between gram-positive and gram-negative bacteria based on the amount of peptidoglycan in their cell wall. Gram-positive bacteria have thick peptidoglycan, multiple layers of peptidoglycan, whereas gram-negative bacteria only have 1 to 2 layers of peptidoglycan. They have a totally different cell wall structure than gram-positive bacteria. So in this technique, we're going to use two basic stains. We're going to use a mordant known as iodine, and then we're going to use a decolorizer, acetone alcohol. So the first step in the gram stain, after you have fixed your bacteria to the slide, you then add crystal violet and let it sit for about 1 minute. The cells are all going to be that purplish color. You rinse the slide, and then with the distilled water, and then you add iodine. Iodine complexes with the crystal violet, and so the actual crystal violet gets into the cell wall and gets below. It's small enough that it can fit below the or between the peptidoglycan layers. The mordant iodine adheres to the crystal violet, and so now you have this complex known as crystal violet / iodine. This is too big to be removed effectively, but with an acetone wash, you're getting rid of some of the fatty membrane, because acetone is a nonpolar substance, and so it can get rid of fatty substances because fats are also nonpolar. And so it helps to get rid of some of that cell wall structure. And in doing so, it also can get rid of the crystal violet / iodine complex from gram-negative bacteria, since they have such a thin layer of peptidoglycan. In the gram-positive bacteria, there are too many layers, and so you're not going to get through them. So then we have the fourth step is the safaranin. So we're going to, after we've used acetone alcohol, we rinse it with distilled water, and then we add our secondary or counterstain. This is known as safaranin. It's a pink color. And so any cells that have been decolorized are going to pick up that counter color, that pink. And so you end up having purple gram-positives and pink gram-negatives. And you can see that right here. And up here is a video on the gram stain. I'm not going to play it because I never play videos in videos, but if you're really struggling, this is something you can look for, is to look for that gram stain video on YouTube. An acid-fast stain is a differential stain that helps to identify bacteria that are gram-positive and that contain a waxy substance known as mycolic acid in their cell wall. And so with the acid-fast stain, we have a primary stain known as carbol fusion, which is going to adhere to the mycolic acid. And then we use a secondary stain known as methylene blue. This technique is a very long technique, a long process. Acid-fast stain is a differential stain that only helps us to identify one type of bacteria, bacteria in the genus Mycobacterium, because Mycobacterium contain the mycolic acid, and so they are acid-fast. So this is a very important stain, again, in medical microbiology, because it can identify bacteria that are acid-fast that cause diseases like tuberculosis or leprosy. In our lab, we have one acid-fast species, and that is Mycobacterium smegmatis, which is acid-fast but does not cause either of the two aforementioned diseases. So in this stain process, we heat fix our bacteria. We're then going to place a piece of bibulous paper. I don't have it on here, sorry. So we place a piece of bibulous paper on top of the slide, and we add carbol fusion to the bibulous paper to keep that slide moist. And then we actually use steam to fix the stain in. After about 15 minutes of staining under the steam, we then rinse the slide off. We decolorize using an acid alcohol, and then counterstain with methylene blue. So the cells that are pink here are all acid-fast. The blue are non-acid-fast. And our last stain technique that we'll talk about is the endospore stain. So this is also a differential stain that allows us to identify endospore formers. There are two different groups of endospore formers that we're typically looking for. Organisms in the genus Clostridium and organisms in the genus Bacillus. So in our lab, we only have Bacillus. So if we're looking at an endospore stain, we're going to use Bacillus to see those little endospores. Endospores are structures that allow cells to stay alive in a dormant form during times when the environment is not favorable for life. So this also uses the steam that we used in the acid-fast stain. But in this case, we're going to heat fix our bacteria, put that piece of bibulous paper over the top of the fixed bacteria, add malachite green, and steam it for about 15 minutes. We then rinse with distilled water and then just counterstain with safaranin. Cells that are endospores are going to be green, or structures that are endospores are going to be green, and cells will be pink. That's the end of this chapter, so I'm going to stop here. I know this was a longer video, and I apologize. Yeah, that's all I can say. You have a wonderful day, and I'll get into chapter 3 shortly. Bye.